In this section, we will talk about the factors regulating or affecting glomerular filtration rate, or GFR. First is glomerular capillary pressure. As we have discussed that glomerular capillary pressure is the pressure exerted by the blood in glomerular capillaries. Glomerular filtration rate is directly proportional to the glomerular capillary pressure. When glomerular capillary pressure increases, the glomerular filtration rate also increases. Normal glomerular capillary pressure is 60 mm of mercury. Capillary pressure, in turn, depends upon the renal blood flow and arterial blood pressure. Second is the colloidal osmotic pressure, which is exerted by plasma proteins in the glomerular capillary blood. Normal colloidal osmotic pressure is 25 mm of mercury. Glomerular filtration rate is inversely proportional to colloidal osmotic pressure. When colloidal osmotic pressure increases, as in the case of dehydration or increased plasma protein level, glomerular filtration rate decreases when colloidal osmotic pressure is low, as in hypoproteinemia. Glomerular filtration rate increases. Third factor is hydrostatic pressure in Bowman capsule. It is the pressure exerted by the filtrate in Bowman capsule. Normally, it is 15 mm of mercury. Glomerular filtration rate is inversely proportional to hydrostatic pressure in Bowman capsule. When the hydrostatic pressure increases in the Bowman capsule, it decreases glomerular filtration rate. Hydrostatic pressure in Bowman capsule increases in conditions like obstruction of urethra and edema of kidney beneath renal capsule. Fourth factor is constriction of afferent arteriole. Constriction of afferent arteriole reduces the blood flow to the glomerular capillaries, which in turn reduces glomerular filtration rate. Fifth factor is constriction of efferent arteriole. If efferent arteriole is constricted, initially the glomerular filtration rate increases because of stagnation of blood in the capillaries. Later, when all the substances are filtered from this blood, further filtration does not occur. It is because the efferent arteriolar constriction prevents outflow of blood from glomerulus, and no fresh blood enters the glomerulus for filtration. Sixth factor is systemic arterial pressure. Renal blood flow and glomerular filtration rate are not affected as long as the mean arterial blood pressure is in between 60 and 180 mm of mercury. Due to the autoregulatory mechanism, variation in pressure above 180 mm of mercury or below 60 mm of mercury affects the renal blood flow and glomerular filtration rate accordingly, because the autoregulatory mechanism fails beyond this range. Seventh factor is the sympathetic stimulation. Afferent and efferent arterioles are supplied by sympathetic nerves. The mild or moderate stimulation of sympathetic nerves does not cause any significant change either in renal blood flow or glomerular filtration rate. Strong sympathetic stimulation causes severe constriction of the blood vessels by releasing the neurotransmitter substance such as noradrenaline. The effect is more severe on the efferent arterioles than on the afferent arterioles. So initially, there is increase in filtration, but later it decreases. However, if the stimulation is continued for more than 30 minutes, there is recovery of both renal blood flow and glomerular filtration rate. It is because of reduction in sympathetic neurotransmitter. Eighth factor is surface area of capillary membrane. Glomerular filtration rate is directly proportional to the surface area of the capillary membrane. If the glomerular capillary membrane is affected, as in the cases of some renal diseases, the surface area for filtration decreases. So there is reduction in glomerular filtration rate. Ninth factor is permeability of capillary membrane. Glomerular filtration rate is directly proportional to the permeability of glomerular capillary membrane. In many abnormal conditions, like hypoxia, lack of blood supply, or presence of toxic agents, the permeability of the capillary membrane increases. In such conditions, even plasma proteins are filtered and excreted in urine. 10th factor is contraction of glomerular mesangial cells. Glomerular mesangial cells are situated in between the glomerular capillaries. Contraction of these cells decreases surface area of capillaries, resulting in reduction in glomerular filtration rate. 11th is hormonal and other factors. Many hormones and other secretory factors alter glomerular filtration rate by affecting the blood flow through glomerulus. Factors increasing glomerular filtration rate by vasodilatation are atrial natriuretic peptide, brain natriuretic peptide, cyclic adenosine monophosphate, dopamine, and endothelial-derived nitric oxide, and prostaglandin E2. Factors decreasing glomerular filtration rate by vasoconstriction are angiotensin II, endorphins, noradrenaline, platelet activating factor, platelet-derived growth factor, and prostaglandin F2. 12th factor is renal blood flow. It is the most important factor that is necessary for glomerular filtration. Glomerular filtration rate is directly proportional to the renal blood flow. Normal blood flow to both the kidneys is 1300 milliliter per minute. The renal blood flow itself is controlled by autoregulation. Blood flow is regulated mainly by autoregulation. The nerves innervating renal blood vessels do not have any significant role in this. Now, Miss Ramona will join us, and she will help us to revise the topic of autoregulation, which was discussed in our previous video on Introduction to Renal System.
Hello everyone, I am Miss Ramona. In our last video on Introduction to Renal System, I have explained the autoregulation and the mechanisms involved in it. Let's revise them together. Do you know what is autoregulation? Autoregulation is the intrinsic ability of an organ to regulate its own blood flow. Autoregulation is present in some vital organs in the body, such as brain, heart, and kidneys. It is highly significant and more efficient in kidneys. Renal autoregulation is important to maintain the glomerular filtration rate. The glomerular filtration rate is defined as the volume of filtrate that forms every minute by the activities of the 2 million glomeruli in kidney. Blood flow to kidneys remains normal even when the mean arterial blood pressure vary widely between 60 millimeter of mercury and 180 millimeter of mercury. This helps to maintain normal GFR. Two mechanisms are involved in renal autoregulation: myogenic response, and other is tubuloglomerular feedback. Let's discuss the myogenic response, or myogenic feedback. Whenever the blood flow to kidneys increases, it stretches the elastic wall of the afferent arteriole. Stretching of the vessel wall increases the flow of calcium ions from extracellular fluid into the cells. The influx of calcium ions leads to the contraction of smooth muscles in afferent arteriole, which causes constriction of afferent arteriole. So the blood flow is decreased. Now let's discuss the tubuloglomerular feedback. Macula densa plays an important role in tubuloglomerular feedback. which controls the renal blood flow and glomerulus filtration rate. Tubuloglomerular feedback is the mechanism that regulates glomerulus filtration rate through renal tubule and macula densa. This is macula densa, while this is afferent arteriole. Macula densa of juxtaglomerular apparatus in the terminal portion of thick ascending limb is sensitive to the sodium chloride in the tubular fluid. When the glomerular filtrate passes through the terminal portion of thick ascending segment, macula densa acts like a sensor. It detects the concentration of sodium chloride in the tubular fluid and accordingly alters the glomerular blood flow and glomerulus filtration rate. Macula densa detects the sodium chloride concentration through sodium-potassium chloride cotransporter. When glomerulus filtration rate increases, concentration of sodium chloride increases in the filtrate. Macula densa releases adenosine from ATP. As ATP is released, it converts to ADP, then AMP, and then into adenosine. Adenosine acts on afferent arteriole through adenosine A1 receptors. Adenosine causes constriction of afferent arteriole, so the blood flow through glomerulus decreases, leading to decrease in glomerulus filtration rate. There are several other factors which increase or decrease the sensitivity of tubuloglomerular feedback. Factors increasing the sensitivity of tubuloglomerular feedback are adenosine, thromboxane, prostaglandin E2, and hydroxyicosahexaenoic acid. Factors decreasing the sensitivity of tubuloglomerular feedback are atrial natriuretic peptide, prostaglandin I2, cyclic AMP, and nitrous oxide. Now, what happens when GFR decreases? Well, then concentration of sodium chloride decreases in the filtrate. Macula densa secretes prostaglandin E2, bradykinin, and renin. Prostaglandin E2 and bradykinin cause dilatation of afferent arteriole. Renin induces the formation of angiotensin II, which causes constriction of efferent arteriole. The dilatation of afferent arteriole and constriction of efferent arteriole leads to an increase in glomerular blood flow and GFR. This was all about the autoregulation and mechanism involved in it. Now Mr. Jude will again join us and continue the topic of renal filtration.
Thank you so much, Miss Ramona. This was all about this section. In our next section, we will understand equations of renal clearance, renal blood flow, and glomerular filtration rate.